Okay. So, thank you guys for inviting me. I'm excited to get to talk to a bunch of other chemists and biochemists. Um, it's not very often that I get to talk to other plant chemists, so it's really exciting for me. So yeah, I'm Allison Barnes. I'm a postdoc at the USDA ARS, located in Raleigh, North Carolina. And I'm also kind of shared with NC State in my previous postdoctoral lab. I'm currently working on a project called CIRCA, working on improving nitrogen tolerance, nitrogen use efficiency and cold tolerance in maize. And I'm kind of managing all of the lipid biochemistry side of that project, in particular running thousands and thousands of samples. So that's something that we'll talk about a little bit later. But first, I'm going to give everyone a brief background into maize, why it's a great system for what we're studying, and then talk about that 2022 PNAS paper and how it led to all of the things that I'm working on now in CIRCA.
Allison, you might want to switch your views. We can see the presenter view now.
Sorry, what?
The
it's presenter view?
slideshow, yes.
Okay. Is that this button? Oh, nope.
Maybe in display settings.
Yeah.
Okay. Let me try that.
I think you might have two screens and you're sharing the wrong one now.
Oh,
Yeah,
it's good.
I think that's what I was doing. Okay. Everyone can see the slide, just the slides
Yes.
now?
Perfect.
Okay.
Thank you.
Perfect. Now you won't see all of my embarrassing notes about things that I need to remember to say. Okay, so also before I start, I just want to give some artistic credit to a former PhD student in Ruben Rayon Alvarez's lab. Her name was Andy Kerr. She is now a full-time science artist. I think she drew this for us when we were writing our PNAS paper to represent the role of indigenous people in the domestication of maize, the role of phospholipids in membranes, and how maize changes across altitudes. So this whole art is hers. She also drew this figure here, maybe. There we go. That talks about how, as indigenous people domesticated maize, it was moved throughout the Americas and also selected for on multiple different scales. So on the geographical front here, maize was first domesticated in Mexico, and then the indigenous people who domesticated it moved it into the highlands and then up into North America and down into South America. So as people traveled, they moved maize with them. And this forced maize to be domesticated and selected for new environments. Additionally, these traits, something that we'll talk about today, start at a very cellular level and go all the way up to thinking about completely different maize architecture. In particular, when you're moving across these different gradients, you need to change things like flowering time. Oops. And then lastly, across evolutionary time, right? So maize was domesticated from teosinte and then to the maize that we all know today. So as I said, maize was domesticated here in the Balsas River Valley and then moved into the highlands in Mexico. So what's important to note here is that maize was domesticated by indigenous people using the teosinte zea maize subspecies parviglumis. So this is the lowland teosinte. It then, when the indigenous people carried it into the highlands, as they moved into the highlands, it had another crossing event there with this highland teosinte called Teosinte mexicana. This carries many of the traditional highland traits like pubescent stems and also anthocyanin in the stems. And so from there, it then moved north into North America, where it crossed with other traditional varieties and land races and was adapted to these specific environments before moving into Europe, into Northern Europe, where we get these now European Northern Flints. From Mexico, it also moved into South America, where it adapted to highlands separately in the Andes. It also then moved into the Caribbean and across to Southern Europe. So this system is really great for us because we can use all of these different populations to look at how traits have changed, how genes have changed, and kind of track different phenotypes across evolution and across different environments. And one of the reasons why this is really important is because the centers of origin for many different species of crops are not where we're actually growing them anymore. So like I said, corn was domesticated in Mexico, but now, you know, corn is being grown all the way up into the northern US, Canada, Northern Europe, etc. The same principle applies for many other crops. And so we need to think about ways that we can adapt crops to new environments, changing climate, and then also thinking about the center of origin for crops. Can we go back to wild ancestors to identifying new variation? And so what this project that I was working on is kind of trying to balance both of those things by going back to Mexico to look at traditional varieties and land races there, and then also look at how that adaptation changed. So I want to get into a brief, quick background here about cold and how plants sense cold. And so here, it's important to note that for a plant, cold is relative for each species, and that the mechanism of different stress-related genes and proteins can differ. So here we're looking at the accumulation of a specific lipid product called TGDG. This lipid only accumulates after a stress event. And so here we're looking at the amount on the x-axis that accumulates during just a controlled sampling. And then here on the y-axis, the amount that accumulates after a cold challenge. And so here we sampled pretty much as many species as we could get our hands on, and then look to see after a cold challenge, what's the ratio of a fresh sample to a cold challenged sample. And so here, for some species that are very traditionally known as cold tolerant, such as Arabidopsis or pea, a lot more of the stress responsive lipid is accumulating after cold. But for a species like maize, it's nearly 1-to-1, and in fact, slightly more TGDG is produced simply by the stress of sampling the plant. So these are important concepts to keep in mind when we're thinking about applying different stresses, how we're sampling plants, and those sort of things that are going to come in later when we talk about how to sample 1,500 plots in 3 hours without trying to ruin your metabolites. Also important to note that chilling stress, cold acclimation, and freezing stress are all different. These traits are important for the type of work that I'm doing because we're thinking about maize in highlands. And so highland environments typically have lower temperatures, soil with less phosphorus, more UV stress. And so the environment itself, for example, for a lowland plant would be more inherently stressful. And so there are these different categories of stress that we need to consider when we're talking about cold, chilling, or freezing. So the first thing that would be happening would be a suboptimal temperature. For maize, that's really going to kind of be below 25, 22, anything down to about 15 is going to be for the most part reversible. It's suboptimal, but it can still grow. A chilling stress would be between 0 and 15 degrees, with 15 to 10 being moderate and 10 to 0 being severe. So during a chilling stress, we'll get decreased leaf expansion rate, decreased chlorophyll content. A lot of photosynthesis traits will decrease to try to prevent the excess reactive oxygen species production, oxidative damage. And of course, some of these traits, such as tassel sterility or crop failure, are going to depend on the age of the plant in which the stress is happening. Maize doesn't really cold acclimate, but some plants do have the ability to cold acclimate. This is a period of exposure to a low but non-freezing temperature. That's an additional adaptation in which sugar, salt, and proline will begin to accumulate other osmolytes, hoping to decrease the freezing point of the water inside the cell, because this is necessary to survive freezing. Because during a freezing stress, what will happen that's different there is that the water inside of the cell will actually crystallize really in the apoplastic space and outside of the cell, causing all of the water in the cell to be drawn out of the cell and causing a dehydration stress that's actually more intense than drought, and the cell can lose up to 90% of its osmotically active water. And so all of these changes can impact membrane composition. And so that's how we've connected highland plants, cold tolerance, and now lipid biochemistry. So for those of you that are not lipid biochemists, here's a brief introduction into lipids. When we talk about membrane lipids, in particular, I'm going to talk about membrane glycerolipids. So we have this component here, that's a fatty acid tail, and then this component here, that's considered the head group. So the fatty acid tails can have double bonds in them, and the more double bonds, the wider apart the fatty acid tails are, the more space that it takes up in the membrane. So here, if we're comparing here this lipid MGDG that has some double bonds in it with this lipid here PG that has one fatty acid chain that doesn't have double bonds, can see how much wider this fatty acid chain or these fatty acid tails are than these two. And I think that this demonstration, this cartoon kind of shows it a little bit better here. So the lipid composition will impact the membrane architecture. So lipids have different shapes, particularly if you have a whole bunch of one lipid together, it may not form a bilayer on its own in the way that we typically think of membranes being formed. So some lipids have this triangular shape here. They're known to be membrane fluidizers, but if we think about fluidizing, we also don't want too many of them because that can cause membrane leakage. So these are important for forming curves if we think about, say, the shape of a mitochondria or the shape of a chloroplast. But we really want to sort of balance those out with these membrane rigidifiers, which are these rectangular shaped lipids demonstrated down here with these groups. So during a time of stress, essentially, what a plant is trying to do is balance the amounts of these two shapes of lipids so that the membranes are solid enough to not leak, but not too solid to shatter, which I'll show you here in a minute. And so here, this is another demonstration of how adding in double bonds to the fatty acid chains impacts the shape. So here we have 0 double bonds with 1, 2, and 3, and we go from being this rectangular shape to being this more triangular shape. So lipid head groups and lipid fatty acid tails can be changed in response to stress. So temperature also impacts lipid architecture. So here we're going from higher temperature to a lower temperature. And so we don't want this leakage to happen. This would be like a liquid disordered phase. What we're really hoping for is this sort of region here, where we have a nice ordered phase. But then as we get to colder temperatures, we can start to see phase separations fractures upon thaw, and also these hexagonal two phases, which if you can imagine this being kind of like a cylinder exiting and entering your screen, these sort of formations form as membranes fuse together during that intense dehydration stress. And then what actually kills the plant upon thaw is the fact that all of these membranes have fused and are now torn apart, causing catastrophic cell damage, as opposed to, say, something like the actual cold itself. So that's kind of my brief lipid chemistry background. Does anybody have any questions about that before we get into teosinte and maize? So before we start this, I want to acknowledge the other two co-first authors. So I joined this project when Ruben came to NC State. He had started this project at Longhibio in Mexico, where Carla and Fausto were working on it. I helped work on sort of the end of the project and then helped to write the paper and move it sort of across the finish line, but obviously none of this would have been possible without the two of them starting the project. And so Fausto and I worked on finishing it here at NC State. But I don't want to forget to acknowledge all of the work that Carla did in beginning the project. So the way that this project started was with glycerolipid profiling from 120 different highland and lowland land races. And what those showed was a difference in the ratio of PC to LPC. So PC is phosphatidylcholine. That's one of these rectangular shaped lipids. LPC is lysophosphatidylcholine. It's the same lipid, just with only one fatty acid tail instead of two. And what they did here was look at traditional maize varieties from the highlands or the lowlands, then grew them in either the highlands or the lowlands, and looked at the ratios of PC to LPC. So here, when maize plants from the highlands, so highland varieties are grown in the highlands, they have a higher ratio of PC to LPC than when the lowland land races are grown in the highlands. However, when land races from either the lowlands or the highlands are grown in the lowlands, there's no difference. So that was kind of, well, it was very interesting to us. Something that I'm not going to show here today, just because it starts to get really confusing, is that they did a similar thing with South American lines. There was no difference with any of the lines from South America. So there seems to be something that's different between highland land races and lowland land races, particularly when they're grown in highland environments. And so then what Fausto did was start to sort of track down and figure out what could be causing this. So he found evidence of selection in phospholipid metabolism at both biochemical and genomic levels. So here we're looking at QST. So this orangey red line here represents the sort of normal drift that we would expect to see. And here, so we're seeing evidence of selection here in these highly unsaturated PCs here, as well in South America. But mainly here of interest to us was this PC 38.5. And again, here, these are the lines from Mesoamerica. He also then started to look doing some PCA and found that these components began to separate based on elevation. So here, the blue are highland lines and the red are lowland lines, and then use that to run GWAS, where he got hits for this gene here, which is what we will be following up on with HPC1, and also then with its counter gene LPCAT1. So we'll discuss that reaction here in a minute. But further following up on those GWAS hits, we identified a QTL that explains this PC and LPC conversion. So this here is also an example of why we often do ratios when we present our lipid data in our lab. So here we have signal here for a QTL hit on the PCs. Here is the signal for the LPCs, but then when we take the ratio of PCs to LPCs, that signal becomes much clearer. And here I just wanted to show this nice example of when we grow plants in the highlands. These are PT, so this is the highland landrace grown in the highlands, and this is B73, the traditional sequenced maize line when grown in the highlands. So there's definitely some stark differences in the ability to grow in the highlands. When these plants are grown in the lowlands, they are indistinguishable. So there's no cost to being a highland plant in the lowlands, but there is a cost to being a lowland plant in the highlands. So this QTL and GWAS led us to a gene that we named high phosphatidylcholine 1 or HPC1. It is a phospholipase A1, and it seems to be more functional in lowland environments than in highland environments. So here in Palomero Toluqueño, which is the PT highland line, we have much more PC and less LPC and free fatty acid, which is the product of the reaction. But here in the lowland environments where lines like B73 are grown, we have much less PC and more of the LPC and free fatty acid, presumably because HPC1 is more active. But that is what we wanted to figure out when I joined this project. So here we looked at both recombinant inbred lines that either had B73 at the HPC1 locus or PT, the highland line, at the HPC1 locus. And then we compared those to CRISPR plants that either had a wild type allele at the HPC1 locus or had a CRISPRed allele that caused a mutation that completely changed the protein. And so here what we can see is that these lowland plants look like the wild type and the CRISPR plants look like the highland plants. So this is PCs, again, that rectangle shaped lipid, LPCs, that triangle shaped lipid, and then the ratio of the two with these patterns matching. We also saw similar changes in both here, this is for the RILs, and here this is for the CRISPR lines, with some of the biggest divergences being in highly unsaturated PCs. So the CRISPR line that we made is shown here. So we ended up having two CRISPR lines, actually, one that caused the missense mutation and one that caused an early stop codon. We used both of those. But being trained as a biochemist, I started looking into what could be the impact of the protein, what could be causing it to be non-functional in highland lines. So we started looking at the domains of the protein and found that here, specifically at this residue 211 in the protein, which is in the flap lid domain, which is important for opening and closing to allow substrate in and out of the protein, we have potentially a key mutation. So here in the lowland lines, we have an isoleucine, and here in the highland lines, we have a valine. So this seemed like a good candidate to follow up on. So we then worked with our collaborators at Cornell. I forgot to put Sarah's name here on the slide, but this is done by Sarah Jensen in Ed Buckler's lab, looking at bacteria that have the same PFAM lipase 3 domains. And she looked to optimal growth temperature correlating with that residue 211. And what she found was that bacteria that had a valine at that same residue had a lower optimal growth temperature than those that had an isoleucine at that same residue. Following up further, we worked with our proteomics and metabolomics core to do some modeling and look at specifically where the catalytic triad and flap lid domain of the protein are to see where exactly residue 211 was and how that could impact its structure. So here's the flap lid in this area here with the catalytic triad and active site being right here. So isoleucine 211 is right here. So you could imagine how being right on the edge of all of this and in the middle of the flap lid having a different residue there could impact the function of the protein. So we then wondered, okay, Palmaritolicanio, the Highland line, has a valine, but how spread through all of these differing populations is this valine? So we looked across HapMap to see the SNPs that existed, and then also looked at the varying populations. And what we saw here is that Teosinte parviglumis, which is the lowland teosinte, has an isoleucine at this residue. Teosinte mexicana, which is the highland teosinte, has a valine there. And then this valine is held through some of the southwestern US traditional varieties, through the Mexican highlands, and through some of the Guatemalan highlands. However, when we look at the Mexican lowlands, the Andes or the South American lowlands, they all have an isoleucine. So this supports that story that we know based on archaeology and the movement of indigenous people, that maize moved into the highlands and then moved north and south, and that when maize moved into the Andes, it had a separate highland event where it was adapted to the highlands there. Here we've also separated out based on it either having a valine or an isoleucine at this residue. And here we see Northern European flints, those same Mexicana teosintes. And here we see more of the tropical maize lines. So what this led us to conclude is that HPC1, the PT version, likely introgressed from Teosinte mexicana, and then is maintained in these lines that are more adapted to cold environments. We also found that HPC1 is associated with flowering time. So here we had a whole bunch of different common garden trial locations. Here's that same image that I showed you earlier of the difference between growing plants in the highlands. And this is what the cobs look like. So here's when B73 and PT are grown in the in the lowlands as compared to in the highlands. So what we saw is that actually having the effect of having the HPC1 PT allele impacts the number of days to anthesis. So here we're looking at meters above sea level on the x-axis. And so the higher above sea level that you go, the earlier you flower when you have the HPC1 PT allele. So, and then when you go all the way down to sea level, you flower approximately 1 day later. So this is, promise you, going to connect back to lipids here. But we also then looked at attack-seek data to see other ways that this could all be inherited together, and saw that there's more evidence of selection also, and still associating with flowering time, with days to silking, and some linkage disequilibrium blocks that correlate to this flap lid domain, like we discussed earlier. Okay, so how does flowering time connect to lipids and highlands? So in the highlands, you have colder temperatures, you have less growing degree days accumulating, and so you may need to flower faster. One of the ways that we know that Arabidopsis flowers faster is by binding FT, flowering locus T, to phosphatidylcholine in the shoot apical meristem. Some really elegant work has come out in the past couple of years showing how FT is sequestered away by phosphatidylglycerol, and then when it's time to flower and the conditions are right, it's released, moves into the shoot apical meristem, and binds to phosphatidylcholine. So what we did was looked at ZCN8, which is a maize flowering time transcription factor. It's the homologue of Arabidopsis FT. We produced it into heterologously produced it in yeast, and then purified the protein, and then extracted the lipid from the purified protein, and then ran that on the mass spec, and we're able to identify PC34-2 as being bound to the protein. We also did some modeling again with Peter from the proteomics and metabolomics core. His opinion was that PC36.2 probably modeled better, but 34.2 was what we found, probably because we were producing it in yeast and not in a plant system. So that's kind of where that project ended. And as my NSF fellowship was up and we were starting to think about other projects that I was going to be working on, we started thinking about how can we scale this sort of question and method up to entire populations. So some of these sampling sets, you know, had 120 lines or, you know, 150, 200. Those are all kind of manageable amounts of data to collect and extract and analyze fairly manually. But with these new collaborations that we're working on with Circa, with the USDA, with other scientists and labs, we're starting to think about wanting to analyze populations, you know, in the 700s, in the thousands. And so how can we start to scale those sort of things up? So that's what I've been working on since then. So I just wanted to give you here a quick overview of how we do all of our sampling and sort of what our process is. And then I would be happy if anyone has any ideas to discuss about metabolomics to talk about any of that. So this is a picture of us in the field last summer here. So these are all of our plots. On this day, we sampled 1,500 plots in about 3 hours. We had a crew of 14 or 16. And so the way that this works is that we let the corn get to between V4 and V6. We take all of our pre-labeled, pre-chilled tubes to the field. We have some punchers. We slide the tubes in. We take 5 punches of leaf tissue, which is approximately 50 milligrams. We flash freeze that sample in liquid nitrogen and dry ice. Like I said earlier, sampling and leaf damage can impact lipids, and so we want to flash freeze that sample exactly at the time that we take it. Then goes back to the lab where we extract the lipids and concentrate them. We do also add an internal standard mix during extraction. We resuspend and add a final standard and then run UHPLC MS. So our MS runs are just 2 microliters. We have 2 different mobile phases that use the different characteristics to push compounds off the column. We're using a reverse phase column, and each sample takes about 15 minutes. So a normal daily run, we run approximately 80 samples. So a typical run for us would look like a blank, an internal standard QC mix just on its own, 10 samples, and then we run a pool QC. That's a pool from all of the samples that day. Ideally, our pool would be the entire sample set, but when you have 1,500 samples, it's not really doable. We prefer to reconstitute the samples that are going to be run that day on that day. So our pools only typically consist of the samples for that specific day. And so on and so forth until we run approximately 80 of our actual samples, which then ends up being closer to 90 or 95 actual runs in that actual day. So then we get all of this data from LCMS. This is our relative abundance across our 15 minutes. So one example that I took here is just this peak at 4.26 minutes, corresponds to one of the more prevalent PCs in maize, which can be identified here based on this 184 peak, and it's also its starting mass. And then we're just integrating the area under the curve for this peak. So we're currently using MS-Dial for alignment, compound identification, and quantification. Before, with our smaller data sets, we were using Skyline, but the issues that we came across were that so much of Skyline was manual for us. And when you have 1,500 samples or 700 samples, or, you know, really anything more than the 200 or so. It was just becoming too lengthy for the type of projects that we wanted to do. So this is what our MS Dio readouts look like. We're not doing untargeted metabolomics. We are doing kind of semi-targeted lipidomics. So we have an idea of what compounds we're looking for, at least for sure the lipid head groups that we want. And then we also only want those compounds that match a reference and have an MS spectra, like here. So we're using Oliver Fien's lab retention time library. Our first samples were run at UC Davis at the West Coast Metabolomics Center. We ran similar standards here at NC State and know that the retention times between the two instruments are pretty similar. So we're using, still just using their retention time library and then looking also for these MS2 spectral hits. So then this is kind of like what my output looks like. We're currently identifying about 125 compounds. And then we're checking these pools and our various compounds for excessive variance. We're normalizing to the internal standards, doing statistics, correcting for field plot design, all of that kind of fun stuff. So, like I said, because lipid metabolism is so interconnected, often looking at the value of just one lipid has not been very informative for us. So we can look at this in several different ways. We have, you know, something like here, A, B, C, you know, the letters of the alphabet. We can look at those amounts of individual compounds. Sometimes it's more informative for us to look at the sums of different branches. So say, you know, the sum of the orange or the sum of the green, ratios of the compounds, like I showed you earlier, or then looking at like ratios of the sums of the different branches. So we've kind of done a little bit of all of that. And it really just depends on what the data set is that we're looking at and what the project is helping to answer. So this is the example that I showed you earlier here, where the two lipids on their own give a significant signal, but not nearly as significant as when we run the ratio. And so the population that we're using here, that's the next population in the pipeline, is this population that we're calling B-Zea. So it's B73 crossed to all different Zea species. So there's like 82 total teosinte crosses, along with Zea diploperennis, Zea luxurians. And then we generated F1s, and then back cross those again, and then self those 3 times. So this past summer, we grew 1,500 of those lines. They were all sampled and have all been run on the mass spec. We literally took the mass spec over from Thanksgiving to Christmas and ran all of our samples. In the fall, after it got really cold, well, really cold for North Carolina, we sampled another half of those. So there was a planting in the fall again, and we sampled 750. The hope was to sample the other 750 after recovery, but unfortunately none of the plants recovered. So we were unable to sample the other half there. Those are currently being extracted and will be run in the next month or so, probably. We also have 2 other large populations that are back crosses and selves, with either traditional varieties crossed to B73 or other teosinte varieties crossed to B73. So that's kind of what I'm working on. Want to say thank you to the USDA for funding me, the NSF and NC State for my initial funding. This is the crew that helped us sample all of our plants last summer. This is our field crew from a couple of years ago with my USDA advisor, Jim Holland, and my NC State advisor, Ruben Brian Alvarez. And then these were Whitney and Yu Chun are my 2 main collaborators from the Metabolomics Center at NC State, who trained me on the mass spec and everything that corresponds to that. And then Katie Smukowski-Heil was my other NSF co-mentor, where I worked on some stuff about yeast that I didn't get into today, but basically trying to use yeast to make lipid phenocopies of maize lipid ratios. So, and then additional collaborators from all of those other great institutes. So with that, can I take any questions?
Fantastic. Thank you, Allison. Are there questions for Allison? That was a lot of ground you covered.
Yeah, sorry, I probably kind of went through it pretty fast.
No, I thought it was good.
I couldn't choose just one thing. I felt like I wanted to tell you guys about all of it, so.
I had a couple of questions.
Yeah.
One technical one. Very early on when you did that first GWAS, this was, I don't know, just a few slides in.
Yeah.
Was the trait that you used in that, I guess that trait that you used in the GWAS was in that particular one, was it the ratio of those two lipids, or at some point were you using the principal component as the trait?
I think that GWAS was the elevation.
Oh, I see. Okay, got it.
But I'm not positive. I didn't do that GWAS and I wasn't here yet when they did that GWAS. I would have to look. I do know that we have done GWAS on both the lipids, the single lipids, and on the lipid ratios. We've also recently messed around with trying to do GWAS on double bond index too, using that as a trait as well. So
interesting. Well, please, if other people have questions, don't hesitate to jump in. You can raise your hand. Maybe Elizabeth has a question here. Hello.
Hi, Allison.
hey, how are you?
Good. It's a small question, but if you go back a couple slides to the you were talking about you were correcting after the 1,500 samples, and you mentioned you normalized or corrected for field plot design.
Yes. So
what does that mean?
This one here, this one.
Yeah.
Okay. So
because I can understand like normalizing to compounds
Sure.
and internal standards, but
yeah, so basically the way that works is that like in this giant 1,500 sample data set, approximately 10% of that data set is B73, highly replicated throughout the field. We have our plot design, so we know exactly how the field was laid out.
Yep.
And then we can look for variance in the B73 and say, like, oh, we noticed that this one region of the field has really weird values in B73. We can then work that into our model to correct for changes in the field that are either due to management problems, weed pressure, soil differences, any of that kind of stuff. These are the things I'm learning now that I'm in like a breeding lab. So those are the types of corrections that they typically do for things like yield trials and that kind of stuff. So that
makes sense. That feels very applicable to what I'm doing as well. Yeah, thank you.
Yeah, no problem.
Other questions are welcome. I guess I have another one. So the last sort of BZIA project that you talked about is exciting, and it seems like a long time in the making, essentially, to create the population that you're analyzing. What are do you have some anticipated findings in mind that you see coming from this work? I'm just trying to, I don't know, kind of piece it all together.
Yeah, so I mean, I would definitely anticipate there being differences in in like the PC metabolism depending on like the elevation, because we saw that before with the traditional land races. Some of the other things I'm not as sure about. This is where I would be really interested in something else that Ruben's lab thinks a lot about is phosphorus acquisition. And
Hmm.
so they do a lot of modeling and mapping of like soil traits onto like where land races are from and that sort of thing. So any of that kind of stuff I think could come into play too, and probably change certain certain land race phospholipids as well. I think that one of the hopes is also just that we can create this to be a population for the like greater community. Because something that we're working on right now is like making a shiny app where you can go in and say like, here's my gene of interest. I want to know like if you have any teosinte or any lines that have, you know, this teosinte in my gene of interest, right? So then you can start to sort of like request RILs or NILs or whatever, so that other people can also say, you know, look at the genes that they're interested in. I mean, there could be other really cool traits too. Like I just think about lipids all the time, you know, but there's like all kinds of stuff with pubescence and anthocyanin accumulation and waxes and all kinds of like totally different things that happen in highlands as well. So there's that. And then also there's just the fact that you know, Zea diploprinus and Zea luxurians, like those aren't teosintase either, right? Those are completely different species. So there could be all kinds of cool traits out there that are just like yet to be discovered. So I guess it's a little bit of a fishing expedition, if I'm being honest, but I feel like it could end up being really cool.
I agree. Well, it's cool just to think about it. Quentin, do you have a question? Please go ahead.
Yeah, I really liked your graphics on the membrane stuff. So to go back to the beginning of your talk, but I feel like I kind of missed the connection of like, okay, the HPC1 is the locus that really gives this highland tolerance. But like, could you connect that back to like, at a simple level, what it's doing to the membrane to survive the different cold stresses? I could just use a second re-saying of it at a simple level as you can, but I thought your visuals were great, and I kind of want to just close the loop in my head.
Yeah, for sure. No, so I definitely could have re-emphasized that. So basically what HPC1 is doing is taking these rectangle lipids and converting them into these triangle lipids. And so when we go back here to like this graphic, we're thinking about that these triangle lipids are membrane fluidizers, right? So we don't want too many of them in the membrane, or it will disrupt the membrane structure. And actually, if we get like too many of them at once, it can actually be more like a detergent to a membrane and like cause the membrane to break apart. So our best hypothesis, because we haven't done the membrane biophysics, is that in the highlands, it's better to have some more of these bilayer forming lipids to help shore up the membrane during this time of stress, to balance out and prevent leakage. So we don't want this whole fusion happening here, like this happens. We want to stay more in this sort of phase, right? So our best guess is that the reason why HPC1 would be less functional or non-functional is that having more PCs emphasizes having more of the bilayer forming lipids and is then better for membranes to survive stress. Because our hypothesis would be that it's inherently more stressful to live in the highlands because of the low phosphorus, the low temperatures, all of that sort of thing. The other thing too is that phosphorus, being a limiting factor, is also needed for DNA and RNA. Phospholipids are often one of the first things that get mined to be used for DNA and RNA. And so there could also be, maybe there's one less phospho remodeling enzyme because we don't want phospholipid metabolism moving through as much because more phospholipids need to be shuttled off to be in DNA and RNA. Those are kind of a couple of different things that could be happening. We don't know what it actually is.
No, that's really helpful. Yeah, that's what I wanted. Thank you.
Yeah, no problem.
Other final questions for Allison?
I have another question.
Yeah.
For the lyso-PC, when you're looking at the PC to LPC ratios, does it matter if the fatty acid is on like the SN2 or SN3 position? Like, does
No,
that change the fluid? Like
so it doesn't, other than it just tells you whether you have a phospholipase A1 or A2. So like a phospholipase A1 will cleave the SN1, a phospholipase A2 will cleave the SN2.
and plants
so
have corn
in
has both
general,
of those, or
we're often pooling all of those because it can be really difficult to get to the level of identification of saying absolutely yes, this is LPC 16-0SN1 or LPC 16-0SN2. Particularly because your LPCs are going to be smaller. When you're running mass spec on them, you may not get as many of the product ions, and so then you don't have as many mass spectra to match, and then it gets harder to say conclusively, yes, this is SN1 or SN2.
okay, thank you.
Allison?
Yes.
Yeah, hi, nice talk. Really enjoyed it. I have a question. So, general, the take-home message I take here, you correct me. This is this phospholipase, modify the membrane by cleaving, so like changing the composition of already pre-established membrane rather than modifying the side chain. So, which have to be done by a desaturase, not a lipase. So this is your overall key message, right? So if I got it right.
Yeah, that's true. And I had a graph here but I took it out last night. There's actually also this idea that cold temperature correlates with having more desaturation. And that's technically like not, at least in Arabidopsis and in total lipids, that's technically actually not true. So we did like a really quick like evaluation using some other studies and comparing like the desaturation, like the double bond index of low temperature to the room temperature sort of comparison. And it seems that at least overall in like total desaturation, there's not more total desaturation at low temperatures. That's not to say that there's not specific lipid head groups where that is true. But at least in the instances that we found in total fatty acids, it wasn't true. So it seems that yes, head group remodeling is just as important as fatty acid chain remodeling in terms of stress survival.
Awesome, thank you.
Well, thank you again, Allison, for a fantastic presentation. Let's all give Allison one more round of applause here.